So the first step is to get Milano-CoA. So the easiest way to draw this out is just start with your acetyl-CoA, but remember this is attached to the KS subunit. It's the ketosynthesis subunit of this megasynthase. Remember that the protein that synthesizes fatty acid is a gigantic protein that has essentially all these proteins as one polypeptide. You guys remember that? Okay? And then basically what happens is the fatty acids, as they're being synthesized, get swung from one active site to another. Okay? So we have the SKS site. That's S is just for us to know that we have here a thiol coming off of that enzyme. And the first step for this is to use Milano-CoA, which we saw in cholesterol biosynthesis, where we shouldn't have seen it. And this is attached to the acyl carrier protein. Okay. Now, how this was synthesized was basically we used ATP, HCO3, which came from carbon dioxide, and then acetyl-CoA. Okay. This basically activated, okay, the carboxylic acid, or the carbonate here, and then that was attached here at this carbon to get that. Okay. Now what that did was that made this a good leaving group, and now is able to do, I'll draw out the mechanism, you wouldn't necessarily have to do this, but I think it's helpful, is this can shift down, and then this can act as a nucleophile, attack that, okay? And actually what will leave will form a tetrahedral intermediate, but let's just, what happens is basically this gets kicked off, okay? So that would be the net reaction. So then what do we have? We have our acetyl group now has been added. This is now attached to our acyl carrier protein. And then this is going to swing it around between the different active sites. So we could say what's left here, the SKS has left, CO2 has left, okay, and then now, aside from that, it's just the reverse, okay? Because this is biosynthesis, we're using NADPH for the reductions. So the next step. Get this. That turns that into a hydroxyl. Now we get rid of water. Oops, sorry. Release, hydrocase. Okay, and in doing that, we get a double bond. Oops, the color coded it. One, two, three, and then we just have one more step, which is another reduction. again, we're going to use NADPH. Where did we get that from? Pentose phosphate pathway. This is a reductive biosynthesis. So you see how things get tied together. Now we're just going to finish this off. Now what we would have to do is get that put back on. Okay, back onto that keto synthase domain, or of the protein, that would be a domain. And then our ACP essentially leaves. And now we can set this back up there. So now we have here. And then we can cycle this however many times. Let's say we want to make palmitoleic acid. So we would need, we have 4 there right now, we would need 12 more. Okay? So we need 12 more, we would have to do 6 more cycles that way. And then what we would kick out here in the end after we've synthesized everything. Okay. 14. Nah, I'm fine with you. But that's the cycle, okay? However you draw out all these things, okay?
Can you?
But that's the cycle that we'd have to do. These steps here.
Does the last step end on after step 4 or on the next step?
On the very last one.
The very last one. Cause you're kicking out ACP, right, with water.
Yeah, you could kick it off at that step. It usually shows it like going from ACP to SKS and then getting kicked off. But it doesn't matter. I'm not, as long as you know those steps. Okay. Okay. Does that make sense? Does that look right to you? Okay, so that's the basic biosynthesis. Really, the important part is, you know, this process, what's attacking. It's a little different because we first have to activate this. Who remembers where this occurs? So where does fatty acid catabolism degradation occur? Where? Mitochondria. So in the cell, mitochondria. Okay, yeah, and it would be the liver because the liver uses fat, right? We learned about that. Where does the biosynthesis take place? Do you remember? It's in the cytosol near the endoplasmic reticulum. Okay, so it's localized at the ER. So I would say in here, localized at the ER in the cytosol, and this is what keeps it separate. Remember, any kind of reaction that counteracts, you know, if there's a catabolic reaction that counteracts an anabolic reaction, we either have to regulate the enzymes in that pathway so that some are shut off when others are shut on, or we have to do them in different places. That's kind of the strategy. Either you have them in the same place and you regulate key enzymes so that one pathway is off when the other one's on, or you do them in different locations. And this is a case of doing this in a different location. This is done in the cytosol at the endoplasmic reticulum because it's all anchored there at the endoplasmic reticulum. Okay. So we went through that, that's the process. How are unsaturated fatty acids synthesized? Same way, except we add on some enzymes called desaturases. So one, desaturases add unsaturation to saturated fatty acids. So the strategy is make your fatty acid first, then use desaturases. Oops, don't know what happened there. That will come in and put in your degrees of unsaturation. Remember that unsaturation always starts at bond 9, right? So it's always delta 9. Okay, so we cool with that? All right. What? Are you deaf?
Nothing, just all good.
Okay, you're dead. Easy crowd there, please, dead people. No, it's true, what are they gonna say? Anyways. All right, so we've seen fatty acid synthesis, okay? Now, the next thing is cholesterol biosynthesis. So, it's very important. And remember, our precursor for cholesterol is squalene. And it has these isoprene units. There are 6 of them. Okay, 1, 2, 3, 4, 5 carbons in isoprene. There are 6 of them. So, 1, 2, 3, 4, 5. 5. And then you just flip them now. Okay, so it goes the opposite way here. Oh, wait. Okay. There's our 1, 2, 3, 4, 5. Okay, so now what I have to do. That. Again, this takes a little bit of practice too. That should be squalene. If we look at how this is, drawing my isoprene, show you where those are at. See how it's just taking the first 3 and then flipping them. Okay. So that's squalene, and that's the precursor for cholesterol. And then basically, if you work this around, you will get a structure. Structure. So you'll eventually get a structure that looks like this. 1, 2, 3, 4, 5, 6, 7, 8. So this is cholesterol. That's the precursor. Imagine bending that all around, and that's what you would get to. Okay. Okay. So that's cholesterol. So this is the precursor. Now, how is it synthesized? That's what I'm more concerned with. Okay. Are we good with that? I remember. So that's squalene. You guys are super excited. That's more exciting than cholesterol. Okay. So you start with acetyl-CoA. Acetyl-CoA. Basically, we drew this out last time that this will attack here. And actually what leaves is the coenzyme A. And what we put on there is a carboxylic acid. And the way I like to draw this, that methyl group up, hydroxyl down. Oops, I did. Remember Britney Spears back in the day? So there we go. We get our HMG-CoA. That's all you have, HMG-CoA. And this is a precursor for ketone bodies. And here's the deal. The ketone bodies are made in the mitochondria of the liver. Okay. Here, we're synthesizing cholesterol, which happens at the endoplasmic reticulum. Okay. So even though this is an intermediate for other pathways, what segregates it going and becoming ketone bodies versus it being used as cholesterol is where it's localized. Okay, HMG-CoA, right? And now you can start to see 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, okay, 6 carbons. We should have 6 here, where 1, 2, 3, 4, 5, 6. But isoprenes, remember, only have 5. So isoprenes, remember, I'm just going to draw it up here. 5 carbons. And we, of course, this is the one I like to start. That's going to leave eventually, right? That's a good leaving group. So this is where we're trying to get to. Something that looks like that. We've got 6 carbons. The next one, reaction, is done by HMG-CoA reductase. Which brings in NADPH. It's a biosynthesis. We get NADP plus out. And what it does, okay, is it releases this and makes this alcohol. So H-CoA leave. And now what we get, and that just makes this alcohol. This is called mevalonate. And so this is the first committed step, or the first, committed step of cholesterol biosynthesis. So that's why this is important. And this is what statins target. They are competitive inhibitors to HMG-CoA reductase. Okay. So now what I like to do is, there are 3 steps, but I just like to summarize it. in one fell swoop. You basically put in 3 ATPs and you get 3 ADPs out. And what we phosphorylate are 2 positions. We're going to phosphorylate this position with 1 phosphate, and this position we're going to put pyrophosphate on there, so 2 phosphates. So what we will get here, 2 minus. And of course I'm color coded for you, but you don't have to do. Okay. And the purpose of this was, 1, we're going to put another good leaving group here. We can't get rid of this hydroxyl and rid of this carbon here unless we have it coupled. And what this does is this makes 2 really good leaving groups. And this was activated for later when we actually put the isoprenes together to make geranyl phosphate, farnesyl phosphate, and eventually squalene. But you don't have to know that synthesis. I just want you to know, how do we get to the precursors that make isoprene units? And this is what we're showing here. So what happens here, I'll draw the arrow pushing for you. So the electrons from here would shift down. These electrons would shift over, and that would kick off that phosphate as a leading group. As the electrons flow in that direction, electronegativity, we break that bond. That's really good driving force, right? Breaking phosphates gives you driving force. Some things that we've learned all the way around. So what do we get here? We get something that is looking a whole lot like an isoprene unit. And there is an isomerase that will flip this. And this is one of the precursors. And then there is isomerase. It can basically go the way I'll show you. That will flip that bond so that here. And now if we look up here, so we need both of these actually, okay? But if we look up here, we can see now in this one specifically, that isoprene, this is just an isomer of it. So that's done by an isomerase. Then what you can do basically is now you can take these 2 components and start to string them together. Basically, this can act as a nucleophile. It can attack here, kick off this pyrophosphate, and then we start to connect them together. But these are the precursors for squalene. So these are the reactive precursors. Greens go right. On to squalene. There we go. So y'all can do that. Again, I apologize for making this Milano-CoA when I originally drew it out. But same reaction. Okay, y'all can do that. So this is what statins target. Statins are a competitive inhibitor. Okay, they mimic actually parts of NADPH. But anyway. So that finishes up that chapter. I don't think there was anything else I really talked about in that chapter. Oh, except that cholesterol is a precursor for what? Basically, all your hormones, right? So I'm just going to go back a slide. Why is cholesterol important? Well, membrane fluidity, which we talked about, but it's also the precursor for hormones. And the hormones are usually more polar, so they get oxygen added, and then usually you get shorter alkyl chain. So usually what happens when you make these into hormones is it takes this, it will hydroxylate it, and then it will cleave this part off because that's really hydrophobic, makes it go into the membrane. And hormones are used for signaling, so how is that molecule that likes to stay in the membrane going to signal? Well, you've got to make it more polar. Not that it's polar, it's more polar. Okay? So does that make sense? Yeah. Shorter alkyl chain. Alkyl chain. Yeah, that looks pretty crappy. So here I'll summarize it. Usually that gets chopped down, and then we start getting hydroxyls at other places. And that was what we talked about. So a person that has high cholesterol, probably their hormones are going to be jacked up all over the place because they have a lot of it present. And so that's something to check. Does that make sense to everybody?
So that top long structure is squalene, and then that is
Yes,
how
this
it's
is
synthesized.
squalene, and then I think linosterol is what you get after squalene. I didn't show that, but it takes like 30 steps. So this is like 30 steps to get from squalene. First you go to linosterol, which cyclizes it, and then in 27 steps, so approximately 30 steps from squalene to cholesterol.
And
This
then
is
that
pretty
thing you
intense.
drew out right after this was the synthesis of squalene.
The synthesis of the precursors for squalene.
All right.
Then these will be made into squalene. Okay, cool. Okay, so that was chapter 16. Then chapter 17, the main point was we're introducing nitrogen metabolism. So let me summarize it because I know it gets a lot of details. Let's summarize the main points so we don't miss the mountains from the molehills, okay? We have to, one, where does nitrogen come from? Two, how is it integrated into carbon molecules? 3, how is nitrogen metabolized? And we can think about catabolic pathways. 4, how are amino acids metabolized and synthesized? That's really what the whole chapter was about. Where does nitrogen come from? How does it get integrated into carbon molecules? Because we know ammonia is toxic, okay? Then the question was, how is nitrogen metabolized? That's the urea cycle. What do we do with excess nitrogen? Why would we have excess nitrogen? Because we've catabolized amino acids. And sometimes we want to synthesize amino acids. So we usually always talk about the catabolic pathway first, and then the synthetic pathway. Okay? And that's what made this kind of chapter a little more difficult. So let me go through each one of these kind of in a stepwise fashion, and we'll see how this works out. Okay? Where does nitrogen come from? From an enzyme called nitrogenase that are in these bacteria, these soil microbes that associate with legumes usually. So we have here nitrogen, N2. Remember what that means, N2, nitrogen triple bonded, right? And the nitrogenase is able to effectively bring electrons in, has a special, what we call FeMoco, iron molybdenum cofactor, that is able to reduce this to 2 molecules of NH3. Now it's expensive, it takes a lot of ATP. So this takes a lot of ATP, our reduced cofactors, NADH, things of that nature. It's expensive, but this is how it actually gets integrated. So this, in your review, I would call this inorganic nitrogen. This here is inorganic nitrogen, has not come from a carbon molecule. So this is how inorganic nitrogen now can be incorporated into carbon molecules. So from here, this can get integrated into things like amino acids, into nucleic acids, so on and so forth. Now this can also be used by organisms. They can oxidize it, use it for energy generation. I'm not going to look at that because we're not those things, okay? So nitrates, nitrites, those things are very important in plants. They are very important in bacteria, but this is what we want to know. Okay, where does that go? Okay. How is nitrogen incorporated into carbon molecules? It's incorporated predominantly through glutamate, okay? So we bring in alpha-ketoglutarate. So the main pathway, bring in alpha-ketoglutarate, 1, 2. And this is drawing a little different than I do amino acids. But I want to note here that this would be your alpha carbon. Okay. So you have a glutamate dehydrogenase that will actually integrate nitrogen in here. So we bring in here DH plus nitrogen. Okay. And then what we get out on the other side here is that oxygen here will leave as water. Okay. And then we get our amine added. Plus water plus NAD plus. Okay. So that was one of the main ones. Remember, glutamate is one of the main agents of carrying nitrogen around. Okay. So we can see that that is there. Another one is glutamine. Okay. And this one uses ATP. So we can go from here. And then we can go a step further. And instead of that, we get NH2 here. And we get water leaves. So a little different strategy. But now we have here call this glutamate. This is alpha ketoglutarate. And this is where the main molecules, the entry point of most nitrogen, comes in through glutamate and glutamine. Okay. There was another one we talked about, asparagine, aspartic acid. Actually, aspartic acid gets its nitrogen from glutamate. We'll see how aspartic acid is synthesized later. Okay. But this is the main one. The other one that we really focused on was carbamoyl phosphate. Okay. So, how can we summarize this? This is the main entry point for nitrogen. Okay? It's into the amino acids via alpha ketoglutarate. So that's the main entry point. We take carbon molecules that are already in our metabolism in the citric acid cycle, and then we have an enzyme that will start to now synthesize amino acids, and these both serve as nitrogen donors. Okay? So these serve as nitrogen donors for synthesizing other amino acids, and we also saw glutamine when we were synthesizing the purines. Okay? So these are the main ones. Okay. So we good with that? Yes. Now the other one that serves very important, more so in the release of nitrogen, is carbamoyl phosphate. Okay. So carbamoyl phosphate. Wow. Basically, we get HCO3, which came from carbon dioxide. Remember, there's an enzyme, carbonic anhydrase, takes CO2 plus water. So ultimately, when I draw this out, whenever you see HCO3 or CO2, they are usually used interchangeably. Plus 2 ATPs plus NH3. Okay. And then this NH3 is used to make this. Carbamoyl phosphate. Bam. Now when I put NH3, this is where it gets kind of confusing and hairy in the book. This comes from glutamate. And we see that when we start looking, if you look closely at the urea cycle, and when we start to tie the metabolism together, generally this nitrogen is being transferred to this from glutamate, but we just generally put NH3. It's getting accepted. You have lots of places that the nitrogen could have originated from, but it's usually transferred from glutamate. Okay. So those are the three main entry points of nitrogen: glutamate, glutamine, and then carbamoyl phosphate, but it still gets its nitrogen from glutamate typically. Aspartic acid and asparagine, which are other entry points, again, they usually are getting that from glutamate or glutamine. Okay, so now we have seen this. How are they integrated into carbon molecules? Okay, they're integrated into predominantly alpha ketoglutarate or into glutamate to make glutamine, and that's the main entry point. Okay, then the question is, how is nitrogen metabolized? Well, we can get rid of nitrogen from the amino acids through transamination reactions. Okay. So, let's go here. So let's talk about catabolism. Are y'all okay? You with me? Okay. So you have these amino acids, okay, and how we get different amino acids built? Well, we get ammonia added to different ones, usually through glutamate. Because the book likes to talk about catabolism, this is usually what we say. We have an amino acid that has some R group. Okay. How do we start to get rid of that nitrogen? Well, we do a transamination. We bring in alpha ketoglutarate. That's the best I've written that out so far. Yes, I know. The little things in life actually make me very happy. Where I can draw a word legibly. Okay, so alpha ketoglutarate, so you should know that structure because we've already learned that. 1, 2. The first thing we do is a transamination. So we have a PLP, pyridoxal phosphate dependent enzyme. And remember, this is able to snatch a nitrogen off of a molecule, pull it off, replace what was there with an oxygen, and transfer it. So that's what we do for this metabolism. We now convert what was a nitrogen there. Now we get a carbon framework molecule that we can, maybe with a few modifications to the R group, shuttle right into our main metabolism pathway. And we'll see that in a little bit. So, and then what happens is we pick it up on glutamate, alpha ketoglutarate, now becomes glutamate. And we can shuttle that and get rid of that nitrogen and make it into something like carbamyl phosphate through basically the reverse reaction of the dehydrogenase. So we can actually take H3. Reverse reaction, and then now we can get off and free up our NH3, or NH4+, I'm sorry. At pH, neutral pH, that would be protonated, it would be NH4+. I'm going to be consistent there. Now, from here, this can actually be excreted by creating carbamyl phosphate. Okay. So we have an enzyme that will synthesize this for us, and that can actually be excreted. So this is the first part. Amino acid nitrogen metabolism. How do we get rid of nitrogen? Well, we remove it from our amino acid that carries it, transfer it first to glutamate. Okay. This then can be shuttled either to glycolysis or the citric acid cycle. And we can get energy from that. But the nitrogen, we're focusing on the nitrogen right now. So we're going to see what happens to that. Well, that gets removed here using the glutamate dehydrogenase. We release that, and now we can use an enzyme to make carbamoyl phosphate, and now we can get rid of this. So this is our catabolic process of removing nitrogen. Is everybody with me so far? Now, how do we get rid of that nitrogen? The urea cycle. Okay. Keep. We enter in some new insert, new slide. Blank. Okay. So that brings us to the urea cycle. We'll come back to the carbon in a little bit. Okay? So let's look at the urea cycle. Ultimately, what we produce in the urea cycle is arginine. So since you've all learned that in the past, we'll just start with that. So we draw 1, 2, 3 carbons, right? Then a nitrogen, then another carbon, and a double bonded nitrogen over here, NH2+, and a single bonded carbon over here. And that is arginine. And arginine is actually kind of the end of the cycle. It's where we actually get rid of urea. So these two nitrogens here are going to give off urea, which then can go to our kidneys and we can urinate it out, get rid of the nitrogen. So what happens here, we bring in H2O, and out the other side we get urea. Okay? Now what that does is it leaves behind this nitrogen, 1, 2, 3 carbons. That's called ornithine. Okay, so this is ornithine. Okay. And then here's where that carbamoyl phosphate comes in. Carbamoyl phosphate comes in right here. So we have our NH2. 3. And remember, that just was synthesized by an enzyme that produced it. It got its nitrogen predominantly from glutamate. We'll see how that all ties together. And what leaves here is the phosphate. So this acts as a nucleophile. We'll attack this carbonyl carbon. Difference in electronegativity. Very good leaving group here in the phosphate. So what's going to leave is that inorganic phosphate. And now we're going to start to build up citrulline. 1, 2, 3. And now what we have here, H2. So you can see where that was added. Draw some arrow pushing here. Release. Is that helpful to see the arrows and see the pushing? Okay, that's citrulline. Okay, now we have to get here, it's only 4 steps to do this whole cycle. We have to get arginine, and we see here, this doesn't cut it, has an oxygen there, so we're going to have to get our nitrogen from some source. Our source is actually aspartic acid, Now, aspartic acid was actually synthesized via glutamate. So, again, the nitrogen's coming from that main carrier. But here's what's going to happen. We're going to bring in here aspartic acid. So what's going to happen here? This is going to act as a nucleophile. It's going to attack this carbon. And that's going to leave as water with the help of some protons, of course. And we'll just put some H2s with that. OH2 is the same as H2O, right? Yeah, I'll draw the movie. What's that? Yes, that's wrong. I should said aspartic acid, but I drew glutamic acid. I'm sorry. So I should stop those. CO2. So then we make what's called arginosuccinate. It's arginine with succinate on it. So the name, learning the names here I think really kind of helps. While citrulline and morphine, not so much. This one, arginosuccinate. So just draw arginine 1, 2, 3. Nitrogen. Okay, and if I want to keep everything, I'll put it R purple. Nitrogen over here. And over here, our nitrogen. Here. Sorry. So what I'll do, kind of show you. Here's our succinate, and of course over on this side, we have our arginine. That side, and that's we have an enzyme that will catalyze the cleavage of this into fumarate. There's fumarate, and then we get back to arginine, and actually we can get back to aspartic acid over here from fumarate. So we can send this to the citric acid cycle. We can make malate. Which can be made into oxaloacetate. Okay. Which can get converted here into aspartic acid. You can bring in glutamate, come out with alpha ketoglutarate. And yet now you can start to see how all that works. Okay? So that's the urea cycle. Okay? Now, one thing I asked in your review is what regulates this cycle? Is that one of the questions I'd asked? And really, it's the enzyme that does this. So the enzyme up here that produces this enzyme here is regulated by glutamate. Actually, glutamate and acetyl-CoA, okay, come together. So, actually kind of, that's terrible. Is that easy? So this reaction here, glutamate is involved in regulating that pattern. Okay, so glutamate with acetyl-CoA come together, okay? And they make a molecule that regulates this enzyme. So that's what's going on. So we good with that? So now we've hit this. Where's nitrogen come from? How is it integrated into carbon? This is the main one right here you want to know. We also sell carbamyl phosphate, and the reason we really introduced that was because we need to understand the urea cycle. How is nitrogen metabolized? We saw how it was taken using alpha-ketoglutarate to make glutamate, and then glutamate gives off its ammonia, which gets converted into carbamyl phosphate, okay? And that we can now excrete urea, okay? Now, how are amino acids catabolized and synthesized? Basically, we've already seen we do a transamination, right? The amino acids, let's first see how amino acids are integrated into the urea cycle, okay? So this is fun. If you don't think so, well, I don't think what's wrong with you. But anyway. Okay. Here we have the liver. I know that doesn't look like the liver, okay? Here we have most tissues. This is it's from your slides, and I know I didn't draw it out, but and then over here we have muscle tissue. Now what connects all these things? Blood, right? So we've got this blood. Okay, how do we metabolize nitrogen? The urea cycle is carried out in the liver, okay? And so what is mostly being done here is glutamate, okay, is getting converted to alpha keto glutarate and back, and the nitrogen is being given off as urea, which can be excreted. So this is where most of our waste is coming through glutamate. Okay, most of the tissues use glutamate and bring in nitrogen, we'll just say NH3, from various sources. So let me highlight what those sources are. Those would be things like amino acids that are metabolized or nucleic acids. Ultimately, those get converted into glutamine, Q, E, and then glutamine is excreted through your blood, okay? Maybe I'll show this as an outflow here, okay? And then what happens in the cell here is that the glutamine transfers its amine to alpha ketoglutarate to make glutamine, and we excrete it here. So this comes in. Okay? And we make glutamate. And then that can go back. Is this okay? And then that can go back through the blood, and we're happy. Okay. Now, the muscle is different it actually excretes its nitrogen waste as alanine. Okay? So what it does is it excretes its waste here as alanine. Okay? So alanine. Okay? Which the precursor for alanine is pyruvate. Okay. And then what happens here? Well, it's going to shuttle alanine out here to the cell. And then what it does is here it transfers it to alpha ketoglutarate to make glutamate. And it gets back out pyruvate, which the liver converts back to glucose through gluconeogenesis. So here in the liver, we do gluconeogenesis. And what we're going to do is kick this back out as glucose and send that back to the muscle. So it gets something back out of it. And these are the main ways that our nitrogen is excreted between the different tissue types. Does that make sense? Okay. Mostly it's being shuttled in our liver through this alpha ketoglutarate and glutamate and expelled as urea, as we talked about in the last one. So we have this coordination between it. Different tissues actually get rid of their nitrogen via different amino acids, but these are the main ones. Skeletal muscle, why would you use alanine? Because you get glucose back out when you send it here, because it makes it into pyruvate, and of course the liver does gluconeogenesis, can kick it back out, and it can reuse it. So it gets some payback from the liver. Is everybody okay with this? Okay, so that's cool. So now let's talk about the carbon, okay, that's still left behind, okay? So we metabolize, that's how we get rid of the amino acid nitrogens. We can shuttle, or from other pathways, we can shuttle them to the liver, and we can excrete them. Okay? Now, how are most amino acids metabolized, the carbon part? They all just get funneled into the metabolic system we already talked about, namely the citric acid cycle, or to pyruvate. So here's how we can think about and summarize what we've learned already. Glucose comes into the cell. 10 steps, gets converted to pyruvate, right? Then we can send this into the mitochondria, decarboxylate it, and make acetyl-CoA. And then now we do our citric acid cycle. And I'll draw just some in a different color. We have oxaloacetate in here. There's oxaloacetate. Okay, a couple steps here. We have alpha ketoglutarate down here. Erase this and make it bigger. Okay. Alpha ketoglutarate we've just talked about, right? You see that all over. Okay. And then we have another molecule here, succinyl-CoA. steps total. Why did I draw it out in this way? Because this is what all those amino acids largely get dumped into. Okay? So, things like alanine, the precursor for alanine is pyruvate, so we just shuttle alanine gets converted into pyruvate by a transamination. So let's think about, just for giggles, having alpha keto glutarate out here, and we get glutamate out, and now we get alanine converted into pyruvate. Cool. We shuttle this amino acid right into the motor that we use all the time. So we can form the molecules that serve as energy into molecules that fit right into our motor. Serine also funnels in here. So we broke it down into these different classes. Serine, threonine, cysteine, they all get converted and dumped in here. What about here? What comes in here? Glutamate and glutamine, right? So the same pathways that biosynthesize things can also be used to degrade things. So we can see running this way, we could start with glutamate. NH2. Okay, which could have come from glutamine. And I'm not really showing it outside the mitochondria. I'm just running out of room. 1, 2. Okay, so we can think about doing transaminations. We remove those nitrogens and we dump them in here. Over here, those pathways are a little more complicated, but things like valine, isoleucine, okay, those get dumped into there as propionyl-CoA gets converted to succinyl-CoA. Okay, up here, things like leucine get directly converted into acetyl-CoA, and also we get this converted into basically ketone bodies. Okay? So leucine we call ketogenic, right? It's the only one that basically is the only one that's totally ketogenic because it makes both precursor for ketone bodies. These other ones are glucogenic. Now, what can get converted into this? Well, that's just aspartic acid. So here, what can get converted into this? Just aspartic acid, glutamine. Okay, they can all get converted into here. So what you see here is the metabolism of the carbon is by converting the amino acids into common metabolic intermediates in the citric acid cycle or glycolysis. Isn't that cool? So we just refine our energy into a molecule that fits in our motor. It's effectively what we're doing. So what do you all need to know? The simple ones. Well, we know how to do a transamination to get aspartic acid into oxaloacetate, right? So those are the easy degradation pathways. Things like tyrosine, tryptophan, those are more complicated. You don't need to know those. So here's the main point too, is that these are reversible, right? The pathways that lead to synthesis can also be used for degradation depending on what we need, okay? And so we can see these molecules as being used to synthesize glutamate or glutamine, or degrade them into these common things. So you see the connection now? Cool. not really, or yes. So that's coming kind of full circle, you see? Okay? It's just like glycolysis, we needed to break things down to get it into our mitochondria, the motor. So we broke down those molecules into pyruvate, into acetyl-CoA. How do we metabolize our amino acids? Our motor runs off acetyl-CoA and these intermediates of the citric acid cycle. So let's just metabolize them, snatch away the nitrogen, get rid of any functional side chains, and make sure everything gets funneled down into our common energy source. And so these you can think of as refinery processes that feed into our motor, the mitochondria. So the easy ones to know, valine and isoleucine, not so simple, but these are straightforward because you already have to know them for the metabolism of forming glutamate. Well, you just use the reverse reaction to get it into the metabolism and then say, oh, it's in the citric acid cycle. So we'll call this metabolism / anabolism of amino acids. Why do I say /? Because some of these things, the catabolic pathway and the anabolic pathway, they use many of the same enzymes. They're using the precursors from these to be synthesized, and they're funneling them in when they break them down. So we had different classes, that's it. Basically, anything on this side of the flow chart, I'll draw this out real quick. So anything on this side of the chart, and something plus what's up here, these are called glucogenic because all of these serve as precursors for glucose via gluconeogenesis, right? Oxaloacetate, that's the first step, right, of gluconeogenesis. You take pyruvate, you make oxaloacetate. So we call all of these glucogenic, and we call this one, or anything that makes only acetyl-CoA or the precursor for ketone bodies, we call those ketogenic. And there's just much smaller class. Leucine is the only one that purely does that. Other amino acids make acetyl-CoA, so things like leucine, valine, they do produce some of this, but they're not purely ketogenic because they only produce part of this. The other part goes into this. So they're keto-glucogenic. They're both. Does that make sense?
So the rest that's not circled is glucogenic.
So all these here that are on this side are glucogenic. On this side, they're ketogenic.
You
So
can
only-
or you can circle the ketogenic.
Does that make sense? I'm putting up the barrier here. This is the wall of impenetrable impenetration. That's a new one. And so anything on this side is glucogenic. Some are also ketogenic, but on this side are the only ones, or on this side, this is the only one that's purely ketogenic, leucine. The other one, there are other ones that dump into that, but they are also glucogenic. Cool. So, what did we cover? How does nitrogen come in? How is it integrated in carbon? How is nitrogen metabolized? How are amino acids catabolized and synthesized? Just through transamination. When we're talking about synthesis, if I tell you, how is alanine synthesis? So let's just do some examples of this. So let's say I say synthesize alanine. Those are the precursors. Okay. And basically, know those, you already know those intermediates, and I've shown you the main ones. Glutamate, alanine, we saw aspartic acid. What's the precursor for that? It's just oxaloacetate. The aspartic acid. So you draw pyruvate for CO2 minus on the end there. Let's do a transamination. Bring in glutamate. We have alpha keto. Glutamate. And now we have aspartic acid. Now remember what's doing these transfers. Paradoxal phosphate, PLP, does the transamination. Those PLP-dependent enzymes that are able to snatch away nitrogen from glutamate, takes it away, kicks out alpha-ketoglutarate, and then transfers it now to our carbon oxygen precursor. So that's all you need to know from that really, if you want to know the bare bones. That's it. But you see how it all gets integrated now in here? We've got it integrating into that pathway, the intersection of what we learned about in chapters 12, 13, 14. Okay. So, see the synthesis, we've seen the degradation, we're good. Those were the main points. Okay, so then we went from there into nucleic acids, right? And that's where we ended, right? We don't need anything new, just nucleic acids, okay. So, the main point of this was that our nucleic acids form from the intersection of two pathways we already talked about. Carbohydrate metabolism and now amine metabolism, right? So nucleotides are the convergence of carbohydrates, meet amino acids to make nucleic acids. This is the deal. This is the bridge between the two metabolisms that we've been talking about. Okay? And so we know that the carbohydrates go through the pentose phosphate pathway. And the amino acids, okay, carry the nitrogen. Carry / harbor nitrogen. And so we have our nucleic acids. Okay, you should know your main purines, right? Have this basic structure. Pyrimidines have this basic structure. So the precursor for the purines has this basic structure. Okay. It's UMP actually. So this is our precursor. And our purines are over here. Purine precursor is inosine monophosphate. adenosine, PI. Over here, it's actually UMP, uridine monophosphate, so don't forget structures that you learned last semester completely. Okay. So, UMP, uridine monophosphate. Okay, so those are our precursors, and of course from this we can get ATP, GTP, and all the other forms of that. And then remember that from these you can get your deoxy ATP, you can get your deoxy GTP, and rely on saying ATP over and over again. And from here, you can get your CTP eventually. Or to methylate the thing, you can get your methylate and deoxygenate. You can get your I don't know why we would have to call it this, but I would call it that just so we know. Get our DTTP, and from this you can get your DCTP. Remember that we produce the ribonucleotides first, and then we produce the deoxy second, right? So that's what we'll go. So we make ribonucleic acids first, second convert to deoxy ribonucleic acids. So that's the strategy for the synthesis of these. All right. Let's do the simplest one first. Okay. Let's do the simplest one first. So let's know where the nitrogens come from. Let's see the synthesis. That's why I have you draw this stuff out. I want you to see where does that nitrogen come from. It's not magic. It's not mysterious. You know, it just doesn't pop inside the blob cell and it gets spit out. It comes from very unique chemistry, very elegant chemistry, in my opinion. Simple and elegant at the same time. So let's look at the pyrimidines. Okay. So what we start with is our nitrogen source is going to be carbamoyl phosphate and aspartic acid. So carbamoyl phosphate, sorry. And I like to draw out this way. And aspartic acid. And I'm okay if you don't put stereochemistry, okay? For this kind of stuff. So we have here 1, 2, 3, 4, 5, 6 membered ring is what we're going to make, and they're all present already. Okay. We have a phosphate. That looks like it's the best leaving group. So we draw this. This leaves. Oops. There. And that's the beginning of making the pyrimidines. Okay. We now make an amide bond, so just like a peptide, bond is an amide. Sorry. We have that. Okay. So we have all of our molecules that we need here, don't we? Let's see, did I flub something? Okay. 1, 2, 3, 4, 5. 6. I'm missing one. I'm flubbed. That's good, we covered at this stage. Okay, now it looks good. And now we can see if we seal this thing up, okay, that's gonna close it up. And where are we trying to get to? Okay, we're trying to get to something. I'm gonna draw it out the best way, but it's okay. We have no worries.
It's connected to
Then what we're going to do
over there is connected to carbon.
is we can seal this up.
You see the plan? So that CO2 group just to go above the carbon.
So we want something that looks
So
like
on this side, the CO2
this normally, but now I've
over
rotated
there
it
it's
a little
connected
bit.
to carbon. Over here it's connected to nitrogen.
This is where we're trying to get to.
Oh,
You
yeah.
can see
I think
right
it's
here.
wrong.
I've just rotated this a
Oh,
bit.
there
So
you go.
here's our
There you go.
nitrogen here. And then here's our other nitrogen over there. So here's our in between. Okay, so then let's just seal this up.
It's right here.
Yeah. That's wrong.
Oh, yeah. Wait.
Oh, you guys, tell me.
So that CO2 group,
Yeah,
is it
yeah,
supposed
that's
to be,
not supposed
okay.
to be there. Yeah, you guys, tell me something.
So I didn't, I was like, I didn't know why it was connected to nitrogen.
Okay,
So
this is
confused.
looking much better. So we have that. Okay. And then basically what we're gonna do is we're gonna form a double bond here. FAD, get FAD, H2. CO2. And now we put the ring structure together, now we can add it to PRP. I can draw out the PRP for you here in a second. PRP, P is paradox, or is 5 prime, or phospho, what is it? It's pyrophosphate. It's 5 prime, phospho, ribose, pyrophosphate. So it looks like this, PRP. Looks like this, it's the mono version here. And then over here, remember, it's PO, and that's our pyrophosphate. Ribose, that's what PRP is. Put in there. Now what we do is we get that added. This acts as the nucleophile. Will attack there. And these leave. And then we synthesize now. Sorry. rid of that CO2 group. And then we get UNP, so CO2 leaves from right here. Phosphate. And then if we do a transamination here, we get CTP, right? So about bringing in GLN, bring out GLU, and then what we can do is just add now our cytosine. So you can see where they're all coming from. They're coming from essentially all amino acids because we saw that carbamyl phosphate, glutamate also sends its nitrogen over there, right? And that's how that's synthesized. So this is pretty simple synthesis. If we want to do purines, that's a little bit more involved. I won't put you through that for the exam. You're welcome. So at least know this one, okay? We can do the purine one, I can do it for you.
Just make extra credit.
But it's a little more involved. We start with PRP, put in nitrogen, but this is what I would like you to see, that this is very simple and elegant. It's amazing that you get this from what you look at the beginning. It's like amino acids and carbamyl phosphate, where the heck? But it really is there, and it's a very simple synthesis. PRP, remember, is synthesized by taking off the bases. So where does PRP come from? If you have an amino acid with a base, or not amino acid, sorry, nucleotide, nucleic acid, with a base here, all you do is bring in ATP. What leaves is the base and AMP. So this pyrophosphate there replace the base. And so this is where we synthesize PRP. Okay. Now then there are all kinds of things that we went over, like salvaging versus synthesis, right? So you can salvage and reuse bases. You can salvage and reuse nucleic acids or nucleotides or nucleosides. You can we can salvage those and renew them, or we can biosynthesize them. So those were the two pathways. The purines, remember, get metabolized, and if they are degraded, they're degraded into uric acid. And so people that have gout is because they get a buildup of uric acid that crystallizes out generally in their joints, and then they get inflammation and it hurts. I know my brother has it. And it has to do with, not properly clearing out uric acid and metabolizing it properly. That was the main thing. But was there anything else in that chapter that I had in the review that I thought was interesting, unique, useful? But those were the main things we went over. Now you can see the integration of what you learned: carbohydrate, lipid, mostly just carbohydrate, right? And how that gets integrated. And it's really cool as you start putting it together, you can see how these pathways start to come together, and from them you start to make unique molecules that are very different from what their precursors are, right? Nucleic acids form a very different function than either the proteins or the carbohydrates, but together now they make this unique information molecule that carries out a lot of functions in the cell.
What about the de novo pathway? What is that?
Which one?
The de novo pathway?
The de novo? This is the de novo
That's
pathway.
de novo.
Yep, the
And
biosynthesis.
So salvage is the other way?
What's that?
Salvage?
Salvage is the other way. That's where it basically just breaks down the DNA into the nucleotides or nucleosides and then just reuses them. Reactivates them. At this step, it would have been broken down. The base is perfectly fine. The sugar is fine. Let's just activate our 5' position and then we can just recycle it. So that was the main thing there, is that these are recycled or they can be biosynthesized de novo. Again, we're going to get lots. So let's take a step back. It's kind of cool. Where does this aspartic acid come from? We learned this from the last one. From where? Where is its origins? Aspartic acid. The citric acid cycle, oxaloacetate, or gluconeogenesis, right? So when we have a lot of glucose in the cell, we have high glucose energy, right? What are we going to have? Lots of oxaloacetate, and we're going to produce more bases, we're going to produce more aspartic acid, and then we're going to start biosynthesizing more nucleic acids. Why? Because we have a lot of energy. We're going to grow and divide, and we need more bases. We need more nucleic acids. So you can see when we have a lot of stuff running through the citric acid cycle, glycolysis, that means we have a lot of energy. That's going to be a pro-growth state. So during that pro-growth state is when you spend that energy to grow and divide. And of course, that growth and division is going to require more nucleic acids. So we're going to upregulate when we get, say, an insulin signal, like we were talking about today. We get an insulin signal, what are we going to do? Upregulate pathways that synthesize nucleic acids, that synthesize amino acids and proteins, because we have energy in the form of molecular energy to spend, to grow, and divide. See how it's starting to fit together? So we learn these as segmented individual things, but you got to start thinking of the big picture. Start putting it together. Oh, I can see insulin would tell the cell, we got a ton of chemical currency around. Let's start building some stuff. And they're going to start building more cells, right? So we're going to need more bases, more nucleic acids. Let's start growing and dividing and proliferating. Coordinated by signaling, which we'll talk about on Friday. How do we get the signal? Does this all make sense? Hopefully it's useful. I feel like in this kind of setting I can expand a little more than what I can in the lecture. Hopefully it's helpful for you. But sit down and think about it after you take the test and memorize all this stuff, right? Sit down and just sit back and think about how do these all fit together and the dynamic interactions that are occurring in the cell that make you survive. It's pretty wild because this is just the surface. All right. Does anybody have anything else to talk about? Want to talk about your feelings or anything like that?
This test scares me.
Okay, it does. It's okay. It'll be all right. Think about Bob Marley. Everything's gonna be all right. He's dead now, so everything's fine for him, I guess. He probably smoked a joint and everything was fine then too, so don't use Bob Marley as an example. Anyway, if you have any other questions, I'd be happy to field them.